Good morning, students. Welcome to my channel GRP Botany. Today, our class is Structure of Proteins. Structure of Proteins. So, protein is a building blocks of amino acids. So, amino acids, building blocks of protein. You know very well, inside the cell cytoplasm, many amino acids are present based on variable group of amino acids. But only 20 amino acids are involved in protein synthesis. An amino acid is considered as a substitutes of methane. Substitutes of methane. So such a carbon, the 4 valency of carbon is fulfilled. Hence it is considered as a alpha carbon. Alpha carbon. Such amino acids is called alpha amino acids. In between are 2 amino acids. The peptide bond is present. The peptide bond is established through dehydration. First amino acid, carboxyl group, and next amino acid, amino group. In between, through dehydration process, the peptide bond is established. In the same manner, n number of amino acids are interconnected one by one. As a result, a polypeptide chain is formed. Such a polypeptide chain is called protein. A polypeptide chain is called protein. So, protein is considered as a macromolecule, that means more than 10,000 Dalton molecular weight is present. It is present in acid insoluble fraction. Such a proteins, as per biology, present in 4 forms. For example, based on the structure, these proteins are classified into 4 types. Number 1, primary structure of protein or primary proteins. Structure of proteins or primary protein. So just the primary protein structure, you can imagine like a straight line. Like a straight line or linear line, you can imagine primary structure of proteins. So n number of amino acids are linked and form into a linear line. And this line, the first amino acid is represent N terminal present in left side, and last amino acid is represent in C terminal, it present in right hand side. So CN terminal parts are present in proteins. N terminal represent first amino acid and C terminal represent last amino acid. So in between, a number of amino acids are interconnected to peptide bonds. As a result, polypeptide chain is formed. Such a linear line of polypeptide chain is called primary structure of protein. It just helps to identify amino acids and their exact location or placement. But the primary structure of protein is always not maintain rigid nature. That's why at certain areas, it will exhibit some of the foldings or coiled. Such a helical or foldings is called secondary helix. Such a protein is called as a secondary structure of protein or secondary protein structure. These are the secondary structure of protein. So, primary structure of protein exhibit helical structures or foldings. Such a protein molecule is called secondary structure of protein. And third one is, but one of the important aspect in secondary structure of protein is the protein is always coiled like a only right hand direction. So clockwise direction there are coiled. That is the fundamental importance of proteins. Whereas in third one is considered as a tertiary structure of protein. In tertiary structure of protein, the protein molecule is coiled like a hollow woolen ball in shape. Hollow woolen ball. So hollow woolen ball means, you know very well, it will coiled like a zigzag manner. There is no specific order. Such a coil-like arrangement is called as a tertiary structure of protein. At the time of the tertiary structure of protein, the protein molecule is coiled like a woolen ball shape or zigzag manner. At the time, it will created many crevices or pockets. One of the pocket is act as a active site. Active site. It is actual site for binding of substrate to transform into either simple molecule through break down the bonds, and maybe it will convert into complex molecule to make the new bonds. So it is indicating tertiary structure of proteins biologically necessary because they are involved in many biological activities. Such a protein is considered as a enzyme. So which protein molecule having biological catalytic nature or catalytic nature, such a enzymes action present. So tertiary structure of proteins are necessary for biological activities because they act as a enzymes inside the cell. So you know very well, all enzymes are protein, but all proteins are not enzymes. For example, tertiary structure of protein act as an enzyme, but secondary structure of protein, primary structure of protein, they cannot act as a enzyme. It is a one form, one structure of enzymes, but they are not act as a catalytic activities. That is the tertiary structure of protein. The common point of these three structure of proteins is either primary or secondary or tertiary. They made up of only one polypeptide chain. That is the fundamental key and important aspect in three types of structure of proteins. And fourth one is quaternary structure of protein. Quaternary structure of protein. So quaternary structure of protein means a protein is made up of more than one polypeptide chains. All these subunits are arranged like a cube or plate or spherical manner, act as a single unit. Such a type of protein is called quaternary structure of protein. So more than one polypeptide chain is present. All are collectively worked as a single unit is called quaternary structure of protein. The quaternary structure of protein is best example is human hemoglobin, HB. Human hemoglobin is consist of two alpha chains and 2 beta polypeptide chains. In the same manner, human insulin is also a quaterary structure of protein. It consists of only one alpha peptide chain and one beta polypeptide chain. So it indicates quaternary means more than one polypeptide chain is present. It represents like a quaternary structure of protein. And these are also important for biological system. So out of the four types of structure of proteins, absolutely necessary for biological activities in living cell or cytoplasm for the catalytic activities, tertiary structure of protein. Along with tertiary structure of protein and these quaternary structure of proteins also important in biological system. That is the today topic. My dear student, thanks for watching. Like, share and subscribe. Thank you.